赖老师他从小是在加拿大他应该算是小留学生啦，加拿大就学大学，然后一直到PhD 都是在 University of British Columbia 拿到的，然后大概是在2010左右，然后2010之后他有到 Harvard， 还有到GH 那里去做了一段时间的 postdoc， 然后也做到那边的讲师，然后可能是2014、2015回到台湾，他可能在清华先待1-2年，然后之后就到中研院，然后目前是在中研院我们刚才说的原子与分子科学研究所还有在台大这里合并。那戴老师他研究的过程里面得了蛮多的奖项，方鼎也一直有拿到很好的方鼎，主要是他做的领域他molecular bioimaging 就是在 imaging 的领域做到很小的这种能够来做imaging， 然后所以因此以这样子的工具拿来研究cell communication、cell Extracellular matrix 的 communication 都是非常好的工具。那因此拿来研究cancer 的 biology 或者是其他的部分都是非常的好的工具我相信一部分是工具的开发，一部分是以这样子的工具有了之后怎么样做好的 biology 或 biomedical science 的研究，这个都是目前非常好的。那我们欢迎赖老师。
So the talk for today is on multi-resolution imaging of the double-stranded break repairs and extracellular vesicles, and I would like to thank Director Chen for the very kind introduction invitation. So allow us to go on. So this is our lab. As Director Chen mentioned, that we are located at the main campus of National Taiwan University. If you don't know where we are, if you ever get drunk, 因为它这个叫醉月湖要来的话就来这边喝一杯，我们就在旁边。So we're very close by to you guys, and it's actually a privilege to be able to do that. And our lab is focused on bio-nanotechnology and molecular imaging. So we're actually, we have 3 main aims. One is imaging, biology, and its applications. We try to fuse them to have a multidisciplinary approach to address biological phenomena. So in the first part of today's talk is on tracking DNA double-strand break repair dynamics in vitro and in vivo. So I am very humbled to be able to come here and present because I'm not an expert in genomics or genetics such as you, but we're trying to develop tools to allow you to assess the double-stranded break repair dynamics in a rather high throughput fashion. So I think everybody here is very experienced, so please allow me to introduce briefly that both gene therapy and cancer treatment, they are known to induce DNA double-strand break. And for gene therapy's aspect, there are many tools that was developed way before CRISPR, right? For example, meganucleases, zinc finger nuclease, TALEN, and you gotta know like before and after CRISPR came out. I think that was around 2013. Before CRISPR came out, everybody was working on these 3, especially TALEN and zinc finger. But after CRISPR came out, 那个就等于是差1年之后，你就忽然之間看見整個 conference 變形了，就是之前大家都在講得很開心的，全部都往這邊偏。So, but what they do is that they both are known to induce double-stranded breaks. And for gene therapy, for cancer therapy is similar that both radiotherapy and chemotherapy, there are methods to induce double-stranded break as well for cancer treatment. Now, how are they repaired? And again, this is really simple for all of you right here, that there are 2 major repair pathways for double-stranded break. One is homology-directed repair. The other one is non-homologous end joining. So since everybody's an expert here, I'm just going to do it really briefly that if you have a donor template in the vicinity of the break, then it's going to allow incision or modification of the gene of interest into the double-stranded break. On the other hand, for the non-homologous end joining, it's just going to be random insertion and deletion. Okay. So there are ways to detect them because it's important. People start to find out that the way their repair affects how the cells can survive under therapy. And not only that, if you're trying to do gene therapy, you don't want the repair or the broken site to be repaired too randomly because they may induce mutation that can be disease causing. So there are the PCR-based methods such as EMC, enzyme mismatch cleavage, HRM, IDAA. So those are the common ones people use based on PCR. They are also sequence-based, such as the conventional Sanger sequencing. And the more expensive one, next generation sequencing. I don't know about you, but for us, NGS is very expensive. So at that time, we were kind of stuck. It's like, man, is there a way that we can detect the DNA repair dynamics at a faster throughput? And for reporter-based, there are GFP or other fluorescence-based reporter to detect either an HEJ or HDR, but rarely both at the same time. And there are also bioluminescence-based, such as by using firefly luciferase, they can detect either one of them. Now, what we are trying to do is actually to develop a non-invasive reporter that can allow longitudinal tracking of the DNA repairs. So conventionally, people have to isolate, extract the genomic DNA, amplify it, and then do the sequencing. We're trying to bypass all that. So what we did right here is a bioluminescence-based repair reporter, and then we coined the term BLRR. So what this allows to do is right here. We have a Gaussian luciferase. This is a very strong bioluminescence. So I believe all of you have seen firefly, right? So those are bioluminescence. So essentially it's enzymes produced by organisms, and then when treated with a substrate, it will emit a signal. Gaussian luciferase is a type of marine copepod. So when 妈祖蓝眼泪就是有一点像那个东西，很亮, yeah. So it's much brighter than firefly luciferase. So what we did right here is we have a Gaussian luciferase, but it's inactive, okay. And then in the middle of it, we have a meganuclease SCE1 cut site with a premature stop codon. And then behind it at the downstream is that we have a T2A with vargular luciferase. It's another type of bioluminescence that also emits signal when treated with a substrate. What would happen is that when there is double-stranded break, we can actually either have non-homologous end joining. So basically in the absence of a donor template, it will actually just by 1 in 3 chance is going to be shifted to +1. So basically the first reading frame of T2A to allow the expression of vargular luciferase. Now, if there is the donor template at this site, then we can actually have HDR. In this case, the Gaussian luciferase will be shifted in frame, while anything behind it will actually be out of frame. So in this case, we actually have 2 outputs. One is GLUC, which represents HDR, and then we also have VLUC, which represent an HEJ. So this is what it's going to look like if you allow it to be expressed in the cell stably. So BLRR under normal circumstances with no cut is not going to have any signal. Then when it's cut and undergo HDR, it's going to emit GLUC, which is naturally secreted into the medium or blood or biofluid. And VLUC, likewise, is the same. And all you have to do is really nice. You no longer have to do genomic extraction. You can just collect medium and then do what we call the BLRR assay. And the best thing of it is that you can preserve the sample for subsequent analysis. And this actually are really good because you can do orthogonal study to validate your findings. And this include protein analyses or sequencing analyses. And not only that, we successfully achieve monitoring of DNA or double-stranded break repair in vivo in a cancer model without sacrificing the animal on site. So this is actually just validation to convince you that we actually did that okay. So in this experiment, we expressed the reporter in the cells with mock transfection. In this case, the y-axis is GLUC4 change, and we don't see much here. If we transfect it with SCE1, which is a meganuclease, which is going to cut this site, all of it, or most of it, would actually go to NHEJ because the reporter donor template is not present. So as you can see, the signal for VLUC went up. Now, if we co-transfected both SCE1, which is the meganuclease, as well as the donor template, we can see that the HDR signal came up quite well. So in this case, this is a validation to show us that, okay, this seems to work. Then the next thing we started to do is, can this help us identify the optimal guide RNA under the CRISPR-Cas9 system? So we designed 6 guide RNA using both benchling as well as chop-chop. And they do yield somewhat similar efficiency. And then we actually started to transfect the cells with the different guide RNAs plus the truncated GLUC. I apologize that this looks kind of confusing, but it's actually quite easy. If you look at the orange, it's NHEJ. If you look at HDR, that would be the blue right here. This is GLUC signal. And what we found is that guide RNA3, which has one of the highest predicted score, actually did show one of the best HDR repair. In comparison, if we look at the other ones, such as number 2, it has 24.44, then the HDR is actually not very high, meaning that the cut was not very good as assessed by our reporter right here. And we further did titer. So titer is a type of, I don't want to use the word cheap man's way of doing NGS, but it's not NGS. Essentially, you just have to sequence it, and then you run through a logarithm. Then you can actually assess the indel and the HDR frequency. And what we found is that our pattern detected by our reporter is actually very similar to titer analyses. And we're pretty happy, but then we knew that the reviewers would not be convinced. So we went to do the expensive experiment. But before that, we were also trying to see if the reporter is sensitive enough to pick up concentration difference. So in here, we have our 293 T cells stably expressing the reporter, and then we co-transfected with truncated G-Luc plus our guide RNA that we have identified to be guide RNA 3. That was the best one. And then we did both BRR assay normalized to the cell viability, and we also did NGS analyses. And this was what we found, is that as the concentration of the guide RNA plus the donor template increases, we do see an increasing trend of the HDR. And similarly, we also saw increasing amount of NHEJ. This was actually a question that was really killed on our manuscript, because people usually thought if you have a high HDR, your NHEJ should go down. That was not the case. And we actually later found other literature that also claimed the same thing. I think conventionally people thought if HDR goes up, then NHEJ must go down. That's not true. And our reporter actually showed that. And not only that, we confirmed that with NGS. So this is the expensive experiment. 我记得这个好像花30万吧。我知道对大家来讲很便宜，对我们来讲很贵。所以 we did this, and then we found that, hey, look, the trend is very similar. That as if we increase the amount of donor template and the guide RNA, yes, like the trend goes up. So again, see, look right here. The NHEJ goes up, HDR goes up. It doesn't mean that they are inversely correlated. Then what we also did is that we were able to detect as low as 1.23% of HDR using our reporter. And we can detect up to as low as 14.7% of NHEJ. So this is pretty encouraging because most geneticists or genetic engineers are trying to increase HDR. If you can get up to 10 or 20, you can publish really well, right? I think the efficiency for that is so low. And if we can detect 1.23, this is really encouraging because if you're trying to optimize your system, you don't have to sacrifice your cells anymore. Just treat it with whatever you want, gather the median, assess it, you're good. NHEJ, on the other hand, is kind of, I think average you get 20 to 30% NHEJ in the background. So having 14.7 to us is satisfactory for the time being. And we also did the correlation analysis, and we found that if we compare NGS HDR signal to our reporter signal, we can achieve R2 value of 0.9722. And then on the other hand, if we compare the NGS and NHEJ percentage to our VLUC signal, part of BRR, we can also achieve a system coefficient of 0.919. So that was quite encouraging. And then we decided to say, hey, could we potentially monitor the repairs in vivo? So what we did right here is that we have the stable lines expressing our reporter plus the donor template. And then we, on the top experimental group, we infected with SC1 lentivirus containing SC1 meganuclease and subcutaneously implanted into the animal. And what we saw is that the cells that's undergoing HDR is going to express Gaussian luciferase. By contrast, the one going through NGJ is going to have VLUC. This is our control group that do not have any cut. And over time, we collected just the blood. Okay, we're not opening the animal, we're just collecting the blood. And 5 microliter to 10 microliter was sufficient for us to do the reading. And what we found is this, is that from day 7 post-implantation to 21st day, 28th day, especially between these two time points, we started to see a drastic increase. And then again, this is the one that's injected with IC1 lentivirus, so that the donor template and the cut is always happening. And we can see that, yes, there's a lot of HDR going on. And similarly, when there's no cut, we don't see anything with NHEJ, but when there are cuts, then the NHEJ is also going up right here. So this is a proof of principle to show that this is feasible to do DNA repair monitoring in vivo. Imagine if you're trying to do like chemotherapy or radiotherapy study in animals, if you're expressing the reporter, or if you have any like DNA repair modulating compounds that you want to treat to the animals, you can potentially just collect the blood over time and see if the two fluctuate. Because people have already started to see that HDR may help the cancer cells survive when it's under cancer treatment. So this is one field that people are jumping into right now. Now, we were not very satisfied because it doesn't sound too physiologically relevant at the time. So, I want to mention that this work is with a very close collaboration with my colleague. I can tell you about the story about how this project came about. But with collaboration with Dr. Christian Bader at Harvard Medical School and Mass General Hospital, he said that, hey, why don't we try to use glioblastoma stem cells? So this is, as you know, a very aggressive type of brain cancer. And then we transduce these cells and to stably express our reporter. And then we also transduce with both the meganuclease and the donor template. Then finally, we treat them with anti-cancer cardiac glycosides. So this is a group of compounds newly identified by Dr. Christian Bader's lab, that they have found that these glycosides can actually suppress cancer growth. But we were wondering, how did it suppress cancer growth? And we found by using this system, could it be related to DNA double-stranded break repairs? And this is what we did. And surprisingly enough, again, these are the groups of glycoside, be it nanotized C, albane, digoxin, sorry, don't know how to pronounce this right. But basically, with higher dosage, we can actually see the HDR are suppressed when it's treated with this compound. And they are known to be anti-cancer. And NHEJ, again, as I was mentioning to you, does not go down when HDR goes up or goes down. And not only that, we did Western blot analysis to show that RAF51, which is one of the major players involved in HDR, is also reduced when treated with this glycoside. Now, these are U87 glioma line, and this is glioma stem cell-like cell lines that we did. And it goes to show that by using this system, we can potentially map new molecular pathway of the anti-cancer compound. So we're pretty happy with this, and I know that you may not be so interested just yet, but usually when you hear Apple talk, there's like one more thing. This is something that we recently also tried, is that, as you know, they are Cas9 nickase. Basically, they only nick one end. So we thought, could it be possible that we use this to also detect how efficient the nickases are? And what we found is that we can indeed do that. By using guide RNA with different nicking sites, we can actually do a very good job in assessing the HDR or NHEJ repair dynamics, even under Cas9 D10A nickase. So on the left right here is our BORR with a G-Luke assay, which is HDR. We show that by using single-stranded DNA, coding or non-coding, essentially this is our control. It doesn't do much HDR repair. But if you have the truncated donor template, then it's nicely repaired right here. And this is just double nicking. So it's nicking site at 1 and 2, or double nicking at site 1 and 3. And then we saw that by the double nicking, we can actually start detecting HDR quite precisely. And this is just the orthogonal study, the titer analysis, that actually show a very similar trend. By contrast, the control, if you only nick once, which is only one single strand, we don't detect much of the repair going on as well. So negative is just mock transfection. NHEJ, on the other hand, we also saw a similar trend. Only do we see a significant amount of NHEJ repair under double nicking, but not single nicking as much. And orthogonal study with titer also show similar. And we're fortunate that this work is recently being accepted for Nature Protocol. So if you're ever interested in applying this, we're very happy to share the plasmid and reporter with you. There are a lot of finicky stuff because bioluminescence is very prone to oxidation. So we're hoping to share this with you soon. So finally, for this part of the talk, what we were able to do is that for BORR, it's really, really efficient, that it doesn't take much handling time. Once you establish the system, it's very economical for labs such as ours. 我们竞争没有大家那么多，但是就是一个只要0.07块，我就觉得这个买一个便当可以做好几次，yeah. And then detection limit is, as we claim, that is about 1% in HDR and 14% in NGJ. Once you set up your system well, this I want to emphasize. And this is an important part. You don't have to sacrifice your sample. You can do longitudinal study. And I think I'm going to skip this because we have been trying to screen for compound with orthogonal study, did it in Vivo, and this is just a very nice system if you're ever interested in that. Okay, so sorry, I'm going this a little bit fast, but I'm going to my second part of the talk, if it's okay with you. Because I was asking Director Chen, I was like, what am I going to present? Because I actually work on this. But so I want to tell you the story. The last story that I was telling you about was actually a collaboration, right? Do you know how long it took? I think it took us about 7 years. So it was funny. It was a work that my colleague, Dr. Christian Bader, we were both postdocs at that time. 然后我后来就发现说，我们华人讲的就是物以类聚。我跟他就是通常待在 lab 最晚的人，然后我们在那边做实验，然后他觉得有一天在那边做实验，然后我也在那边做，就差不多7、8点了，然后他跑过来跟我说 ，Hey Charles, I want to try this. Do you think it's gonna work? I'm like, sure. And then we just started chatting, and then time goes on. We both became PIs, and we both don't have money. And then it kind of dragged on for a while. And then by the time we got some money, I was like, hey, I think we can pick up. So in fact, we were actually able to create an original reporter in Boston, but the design was flawed. Both of us had no clue why it was flawed. Until one of my students had his project failed twice. I said, hey, look, dude, 你要不要换一个 project？ 是我跟我以前的同事一起创，可是我们都一直卡住，你要不要试试看？他回去1个礼拜回来之后，老师，你们设计里面有3个 premature stop codon， 所以你再怎么样都不会过去。He's actually the one that made the project possible. So 他的名字叫简泽勇。所以 anyway, what I'm trying to say is that 我们台湾的学生其实非常棒。然后刚才前面那个 story 其实真正是他创出来的。So coming to this one is on extracellular vesicle. 那当时我跟 Director Chen 就是说，这个东西好像不太对。后来我想说，他说没关系，只要跟nucleic acids 和 protein 有关，那应该 OK。那我说好，那我讲讲看这一个。So 这个其实中文来讲是我们的本业，呀，刚才那个其实是我们的一个 pet project， 后来就是很 lucky 的可以做出来。So I don't know if you're familiar with vesicles, so I'm going to go over it briefly. People usually use extracellular vesicles or particles, and since I grew up in Canada, I'm kind of like old soul. 所以我很喜欢听垃圾油，小时候很喜欢听那个 radio， 后来就变 podcast。你们以为 podcast 现在夯吗 ？No， 是 podcast 出来的时候，那时候就很夯了，就很久以前的事情。是因为疫情的关系，所以大家才开始听。But there's a show that I used to, I still like to listen to it, but it's a show called Vinyl Cafe with Stuart McLean by Canada Broadcasting Company, and it has a slogan that says, "We may not be big, but we're small." And it sounds a little bit counterintuitive, but I thought it fits Vesicle quite well. 那我其实从小到大都在听这个秀，然后到我在做博后的时候也在听，因为我在加拿大，我在美国那时候会想加拿大嘛。那anyway， 所以对我来讲就是大家会，我比较辛苦一点，就是我又想台湾又想加拿大，所以在想加拿大的时候就听这个秀，然后一直听，就等于是陪伴着我研究的那段时间长大之后，很遗憾的这位主持人后来在2015年的时候就因为我没记错的话是皮肤癌然后过世，所以他对我来讲影响颇深的。你们听可能会觉得这个秀很无聊，因为他讲的都是加拿大人的一些那种故事，然后那种荒野大草原然后什么都是小故事，但是他很有加拿大的一些精神。Okay, so going on. So what are extracellular vesicles? So vesicles essentially are different groups of vesicles released by cells. So if we label the cells completely green, as you can see on the left with GFP, you can see that this is live cell confocal microscopy, and you can see that there are many particles or vesicles surrounding it. If you harvest them and do SEM, you can see the canonical lipid bilayer right here. And they can be coined terms as exosomes, which ranges from 40 to 100 nanometers, or shed microvesicles, they can be bigger, up to 1,000 nanometer, or apoptotic blebs, and oncosome, which people claim that they are cancer-release huge vesicles that can influence migration of the cells. Now, other group also found that developing cells actually release microsomes. So essentially, the cells will have like a pomegranate structure. 就有点像那个中文叫什么石榴嘛，它就是一颗里面含有很多小颗的，然后之后它再释放出来，然后也是在 development 的时候对 cell migration 是有导性的。I miss exomeres. So in addition to vesicles, there are also particles. And then David Leiden's group from Cornell Medical School coined the term exomeres. So they don't have the lipid bilayer. Essentially, they're just particles. And for the longest time, people thought that these are garbage released by cells. But starting in 2000, so what I meant when people thought it was garbage, it was around 1960s. People under TEM, they were able to see things. They thought, man, this is just junk. But starting around 2000, early 2000 to mid and late 2000, people started to find that, wait a sec, these vesicles, be it exosomes released by fusion of multiphysical body to the membrane to release exosomes, or shedding of microvesicles or ectosomes or particles, they found that they all carry bioactive cargo, including protein, nucleic acids, and lipids, and these can be delivered to recipient cells. So the extended analogy I usually use 就是很像我们那个年代有无线和没有无线出来的时候 ，before and after wireless, right？ 大家都还记得黑金刚吗？会点头的就是在我这个年代，不知道黑金刚就是以前那个手机比这个还要大，然后它的蓄电力超过30分钟就很棒了。Yeah, okay. Why do I mention that? Because when I was doing my PhD, I used to work on something called gap junctions. Gap junction essentially is physical channel formed between cells to allow cell-to-cell communication. Vesicle in this case is like a wireless network. I mean, do you remember like 802.11b? Like wireless network card? It really sucked, okay? But Vesicle is a little bit like that. When people started to find out that they can actually mediate intercellular communication, it just completely changed the level of the biologist's understanding in how cells communicate with one another. Because they don't have to come in contact with one another anymore. They just have to release the vesicles. So since then, there's an explosion of vesicle research, especially relating to disease. And this include cancer, neurological, heart, immunity, and infection. I don't want to talk about the pandemic, but anything that you think is disease-like can be related to vesicle. Yeah, I'm talking about literally anything. Like just so many things, there is an explosion of things. But a really hot field for venture capitalists is trying to use vesicle as a biomarker. Why is that? Because what they're trying to do is liquid biopsy. If you can harvest biofluid, like urine or blood or CSF, then they're hoping that they can harvest these vesicles to actually characterize the bioactive cargo within them. And this would be nice because, for example, if you have a cancer patient undergoing therapy, if you can actually monitor it just by extracting the biofluid, wouldn't that be nice? That's what a lot of people are trying to do right now. On the other hand, scientists or preclinical scientists such as are trying to employ these vesicles as an endogenous therapeutic vehicle. Reason being is that I guess everybody knows about liposome. It's still hot right now, but they have a joke within their field. They say that over all these years, they really successfully cure cancers in mice. But unfortunately, once it hits clinical, most of them fail. And to my knowledge, there's only about 3 to 5 FDA-approved liposome formulation. I could be wrong, but humans and mouse just have a very different immune system. So VCs, as well as scientists, are trying to see if vesicle can be used as a therapeutic vehicle instead, because they are endogenously released by cells. Due to time, I'm going to go over this really quickly. So there are 3 main ways. People are trying to use vesicle as an immunotherapy carrier. This had gone on pretty early on, around early 2010, or if not slightly early in late 2000. Yeah, so there had already been clinical trial going on by using vesicle as an immunopresentation medium. People are always trying to load RNAi into the vesicle with targeting effects. So if, for example, here they target, they can actually do decoration with rabies glycoprotein, and they can actually do tail vein injection, and the vesicle, according to them, can pass through the blood-brain barrier to deliver siRNA into the brain. That's what they claim, even though that paper was quite controversial. That was by Matthew Wood's group. It was published in Nature, I think, Biotechnology or Nanotech back in around 2012, I believe. And of course, other people are trying to load basically any kind of compound that can be anti-cancer or whichever into the vesicles to deliver them with targeting effects. That's what a lot of people are trying to do right now. On the other hand, I think people know about regenerative medicine. I think this is also a very hot field. People are right now thinking that vesicles are in fact the actual player in regenerative medicine rather than the cell itself. Because what they found is that when they're trying to implant a mesenchymal stem cell into the animal, the mesenchymal stem cell does not have to be at the site of the therapy. They can be apart, and they later started to find out, wait a second, if I isolate the vesicles from these mesenchymal stem cells and inject them, they can actually achieve a similar therapeutic effect. So that's another field of interest for the vesicle people right now. Now, when I was starting the project, 台语要怎么讲？就是我看不到，就是不太相信那种。你要想，我以前就是用电话是有线的，你跟我说无线，然后可以讲话，是很神奇。So I was like, I want to see it. So there are ways to see it at the time, and predominantly, I would say the golden standard is SEM and TEM. So as you can see right here, these are glioblastoma cells, and you can see all these like little dots on the surface. So this is by Johann Skok, and that was published in my mentor, Dr. Sandra Brayfield's group back in 2008 in Nature Cell Bio. And I think this is one of the pioneering studies that show that vesicles can be released in abundance by cancer cells. And of course, later more technology came out that you can actually look at the physical properties of the vesicles, including dynamic light scattering, nanoparticle tracking analysis, zeta field, ozone. 我没有 conflict of interest， 只是想要跟大家讲，这几个技术都有它的 limitation， 不要把它当作是神来用。So if you're in this field, happy to chat with you. They are not as glorified as you may be educated for their use. So happy to chat afterwards. Other people, such molecular biologists such as us, will try to tag those vesicle markers such as CCC3 with fluorescent protein. Okay. And of course, chemists are trying to use antibodies against vesicle markers such as AD1. So essentially, these are tetraspanin proteins that's often expressed on vesicles, and as well as chemical dyes. Essentially, these are lipophilic dyes, such as PKH dye and lamberite. Now, if there are trainees in this room, I will usually share my experience with you is that to read the instruction manual carefully. These dyes are initially used to track cells. They are not used to track vesicles. So my extended analogy is that if you're trying to color your Easter egg, you have the dyes, and then these eggs are your vesicles. 你最后还是要把它洗掉啊，不是吗？你要怎么把它洗这些 nanovesicle 是一件很困难的事情。And not only that, I think one thing that hit me when I was doing my postdoc is that the half-life in vivo of these dyes is 5 to 8 days and up to 100 days. If the vesicle can live this long in animals, I think there is something really wrong with your body because they should be degraded or turned over. So at that time, I was gonna go, no, it doesn't make sense. Not only that, later reports started to come out that these dye actually automatically form micelles because they are hydrophilic. They have very long hydrocarbon chain, and they themselves can actually gather together to form small, tiny nanoparticles. So that's actually even worse. They give you a lot of false positive results. And this was what I mentioned. You have to get rid of the dye by washing them. And if you have done ultracentrifugation wash, if you're good, each wash you'll lose at least 30%. And they are hard to harvest if you have done it. So at that time I said, okay, I don't want to do that. That's just crazy. Similar principle applied to other lipophilic dyes. So we set a couple criteria at the time. I said, is it possible that I can label vesicles specifically and to yield accuracy in terms of spatial and temporal resolution? And finally, what can I see? And I was like, can I see a whole animal? And I think I was young and reckless at that time. And can I further actually zoom in to see subcellular resolution? And we're very fortunate that at that time we did develop a set of bioluminescence reporter. And indeed, by injecting into the tail vein of the animals, we can actually use bioluminescence to see them, essentially identifying the biodistribution of these vesicles that you administer. And we're very fortunate to collaborate with cancer immunologist, Mikhail Pet's group, and was able to track how melanoma released these vesicles to educate the mouse immune system. But this actually happened over time. At first, there's actually defense by the immune cells, but later they actually broke through that defense and started educating the immune cells for their own use. So that was very interesting story, and it was published in Science back in 2016. Now, on the other hand, we developed a series of fluorescent proteins that allow us to do live cell, and not only that, subcellular imaging of the vesicles. And this work was published in Nature Communications in 2015. So when I came to Taiwan, there's another thing in terms of application. We were also able to use the reporter we developed to invent microfluidics to capture vesicles from biofluids from GBM patients. Yeah, so if you're not familiar with GBM patients, they usually don't have a very long, they don't have a very good prognosis. I think 通常1年到2年就已经算非常非常好的一个 outcome， 所以他们是希望说，如果可以从血液中去萃取出这些 vesicle 来，然后去做genetic subtyping， 或许这个可以帮助那个癌症的医师用不同的方式去帮忙这一些脑癌的病患可以活得更久一点。So that was a paper that we published in Nature Communications in 2018. Yeah, when I was in Taiwan, I was like, 每次用一个 reporter 是可以看到一种 we can only see one, but not both, right? I want to see from vesicle to cells, and then finally organs and the whole animal. Is it possible to do that? And that's actually what I set to do when I came back to Taiwan. So what did we do? What we did right here is actually to use something called bioluminescence, resonance energy transfer. Okay, I believe most of you are familiar with FRET, right? Fluorescence, resonance, energy transfer. Usually there are two fluorescent proteins coupled to each other in close proximity. If one shines, it will actually activate the other one, right? BRET is similar, but instead of using a pair of fluorescent reporters, you have one bioluminescent reporter and then one fluorescent reporter in close proximity. And here we use nanoluciferase, which is much brighter than most of the luciferase you see on the market or on the street. And then essentially, if you treat them with fluorescein, it's a substrate, and in the presence of oxygen, it will emit a peak signal at 450 nanometer. This, in close proximity, will excite EGFP to also emit another fluorescent signal at 510. So this is bioluminescent signal, this is fluorescent signal. So one reporter will actually give you two outputs. And this is actually quite convenient for our use because we tag them to the inner leaflet of the plasma membrane. So when we express them in the cells, all of the interior membrane of the cell will be labeled with this reporter. And likewise, the vesicle released by them will also be labeled. I'll go over really quickly. We label the inside because we're worried that if we express on the outside of the membrane, it might actually create sterical hindrance to other protein on the membrane for the vesicle to interact with other cells or tissues. Reason being is that more and more recent reports start to show that proteins such as integrin on vesicle can actually direct their organotropism. So we're trying to avoid all that just by decorating on the inside. Now this talk excluded a lot of the characterization that we did due to time, but here we're trying to show you that under live cell microscopy, and this is confocal, we can see that we can really witness vesicle being exchanged between two populations. One is the reporter that we developed, we call them POMGRET right here, and the other one is POM-TV tomato, which we published previously. And by co-culturing them live, you can actually see these vesicles being released by both populations and be readily exchanged. So this might not seem much, but think about it. All the cells that we know today actually release vesicle, and they are being exchanged all the time in the tissue, in the organ. So imagine the amount of cargo that's being exchanged. That's something that's really interesting, and I think to grasp that concept is quite amazing, because for example, between you and I, it's not just void. There's a ton of wireless wave. It's just that you don't see it. Vesicle is similar in the body. You don't see it doesn't mean that it's not doing anything. They're actually exchanging a lot of stuff. And this is just a proof of principle to show you that. And beyond that, in Taiwan, we're able to employ something called SURF, or super resolution readout fluctuation nanoscopy. So this is what it looks like under conventional epifluorescent microscope by using SURF. So this is a logarithm-based nanoscopy. We can actually identify, we can resolve these vesicles. So 这边看起来就糊糊 is kind of like blur, but now you can see individual dots. And similarly, you can see the resolution now. So what this allows us to do is to actually quantify them more precisely. So we next went on to in vivo study. So we injected the label vesicle into the animals, and then we injected substrate, and we started imaging them at different time points. And what we found is that under bioluminescence, we can see a ton of vesicle going around the body. So this makes sense, because once you inject something in the body, they will quickly distribute This will be followed by a second phase, which is the processing phase by different organs. And we were very excited to see this at that time, because the previous generation of reporter we generated, signal will die down within 3 to 5 minutes. But this one, we can actually track them for about 15 minutes. And not only that, this is actually the GFP signal that we got by Brett. This is how ugly it looks if you actually use external excitation. You basically can't track much at all. So we thought, this is awesome. I think we can actually do some in vivo work now. And not only that, we did ex vivo imaging, so we harvested the organs out from those animals, and we see the lung actually came up, followed by the liver and the spleen, under both BLI and Brett signals. And this, again, is what it looks like if you do external. So if you want to image nanoparticles, external light source excitation really doesn't work. We're trying to use internal light to actually excite them, because the signal to background noise ratio is so much better when we do that. And beyond that, we can actually section the tissues that you saw earlier from here, and then we actually did immunostaining, and we were able to do nanoscopy on them. And very importantly, we can actually quantify it. This is one of the hardest fields in Vesicle right now, is to quantify the vesicles, especially in vivo or ex vivo. And again, as you can see, we quantified 70 images per organ by using ImageJ, and we saw a lot of going to the lung, and that's what we saw. And then for the spleen, liver, and kidney, all of them match with what we saw under in vivo and ex vivo imaging. Then we kind of go, okay, we have characterized how they work. And due to time, I'm going to jump really fast. Then we decided to see, okay, we know that cancer vesicles have been reported to have tropism. They can actually deliver to their pre-metastatic niche. At least that's what they claim. So we use a liver cancer model, and under a syngeneic mouse model. So basically it means that the immune system is intact. Most of the cancer study, or I should say some of them, use immune deficient mice. And in our opinion, we're trying to have at least some immune system intact to see if the vesicle will be, sequestered or captured by them. So what we did right here is that we use HCA1 liver cancer expressing our reporter, and we orthotopically implanted to the liver. So as you can see, these are the tumor sites. Interestingly, these cancer can actually migrate or metastasize to the lung over time. So we were wondering, could vesicle play a role? Would the vesicles released by these liver cancer go to the lung? And not only that, are there specific proteins that directed them to the lung? And what happens if we actually make them not go to the lung? If we direct them away from the lung, what would happen? And what happens with the distribution over time? And basically, we harvested these cells, harvested the vesicles, and did mass spec. We identified a group of membrane proteins, and we knocked them down. Okay? Is that going a little bit too fast? We knocked down potential protein on the vesicle membrane that has lung tropism. Yeah. And then we injected to the animals with several injections, 1, 2, 3, and 4th injection. So we're trying to mimic continuous release of vesicle over time in the animals as the tumor develops. And what we found is that at half an hour and 70 to 5 hours, in fact, a lot of the vesicle actually went to the lung. So black is our control. This is our scrambled. And then, interestingly, there are a couple protein candidates, such as SLCO2A1 in red, and then CLIC1, and then CT13. When these membrane proteins are knocked down in the membrane of vesicles, they don't go to the lung as much now. Okay, so the y-axis is the percentage of detected signal in the lung. Original is around 60. After we knock them down, it dropped by around 20%. NERF1 just coincidentally served as our internal negative control. It did not affect the tropism. Liver is kind of interesting as well, that the ones that went down in the lungs actually shot up a little bit more in the liver. Okay, and not only that, the spleen and kidney also show a similar trend, that when it went down in the lungs, they actually came up in the spleen and kidney. Heart and muscle, not much of them went there, so we consider them not very statistically significant. But this is actually a breakthrough, because most studies will focus on one organ. They will say that, oh, okay, it doesn't go to the lung anymore, end of the story. But what happened is that the distribution is actually very dynamic at an organ systems level. 它今天不去浪，不代表说它不会跑到其他地方去。大部分的 study 就在那边就停了，它就不再讲。但是其实它会跑到其他去的时候，这个在生物学或者是癌症学的时候很重要。搞不好它在癌症的不同阶段的时候，它的走向是在不同的器官在游走，只是你看到的是结果最后那个方向。So this is what we're able to do and achieve. So again, I don't want to delay everybody's lunchtime, but we were able to achieve multi-resolution imaging with dual output, as well as long-term in vivo tracking, plus really sensitive biodistribution analysis. And last but not least, I would like to thank two of my students, Mr. Jasper Chen and Anthony Wu. They are the two who led the two projects that you saw earlier. I would like to also thank a lot of collaborators, including Dr. Chen Yunjing from National Tsinghua University, and our core and Dr. Zhongqi Liao, and more importantly, many collaborators from abroad, also in Taiwan. I'll put the names right here. And this is the collaborator I was talking about, Dr. Christian Vetter, who made the double-stranded break repair reporter possible. And last but not least, I have to thank all the funding sources, including MOST, as well as Academia Sinica for making our work possible. So thank you very much, and I'd like to entertain any question you might have. Thank you.
So for your first talk, I wonder whether for the education part, I may not have followed your talk like close enough. So is there any sequence specificity for measuring those non-homologous or homologous recombination? I'm trying to lead to one important like a famous development for transposable element modulation. So that would be like endogenous sequence, and I wonder for your reporter system whether you need to turn into a specific sequence, or it's in general you can see the entire homologous recombination event.
That's an excellent question. That's exactly what the reviewer asked. Yeah, no, we cannot. Yeah,
Okay.
I think that's a short answer to that. But if there's, how do I say this?
But we can design like a
Yes.
specific sequence based on
Yeah,
that. Then
you can design expression
Yeah.
cassette to see if your sequence of interest may, however it acts in vivo. But you're spot on. That was one of the major concerns from the reviewer. We cannot detect endogenous sequence. So 它不是万用刀 but 它有它的好处是，如果你今天有 develop 一些genetic tools, you want to see the efficacy, right? Like you want
you
to
actually have specificity, so that's
Right.
actually not a bad thing.
right. So yes, we're very humble that it's not that great, but it has its pros and cons. Yeah.
Yeah, great. And for your second talk, you were kind of trying to link extracellular vesicle into genetics, and that's definitely relevant. It's definitely because for all those cargoes and for all those communications, the cell actually would have like expression and modulation changes after receiving those cargoes. So.
Yeah, indeed. So I don't know. There are many geneticists here, so 我不敢乱讲话. But most of the vesicle nucleic acids are fragmented RNA. Yeah, I think that's contrary to people's belief. Not so much on DNA that has been reported, but mostly non-coding RNA. So the fact that you mentioned transposon and other stuff, that's what is microRNA and all those it's just tons of long non-coding RNA. Those are a lot of stuff that people are looking at right now. And there is consortium in the States, I think it's called EXCRNA. They do have like a position paper to ask people to try to follow in terms of vesicle isolation and the nucleic acid isolation, because there is a very strict criteria that they're trying to set. Because you may be isolating a lot of non-vesicle related RNA, which is actually fine, which is actually okay, but it comes down to what you're trying to claim. Yeah, because I think many companies don't care anymore. They just said that anything extracellular, be a vesicle or free nucleic acid, those are fine, as long as you can map it, characterize it, find a pattern, and then do liquid biopsy. They're fine with that.
That will be for biomarkers. But
Right.
if we are talking about the consequence of the recipient cell maybe have some gene expression changes, then it's better to be extracellular vesicle. And like microRNA, pi RNA, and
Right.
link RNA will be all relevant.
Yes, yeah, but that's really not easy, especially under physiological context. As you may, the reason why we did it in vivo was because most of the study for vesicle is that you isolate something from cell group A, and you smash cell group B with a ton of those vesicles, and you see a phenotype. Those, in my opinion, are not very physiologically relevant. Yeah, I think that's something that the field is trying to move into right now. More in vivo, because in vivo is not easy to do for vesicle study. But the fact that we have developed tools is to help people to see that. It's not perfect because it's a reporter system after all. But to track nucleic acids delivered by them, oh, that's not easy. But yeah, people are looking into it.
It sounds like we may be collaborating on.
Maybe we're doing that right now, so be happy to. Yeah, I think just to add to it, not only are they related to disease, people have found that, say for example, under exercise, muscle cells can actually release vesicles with specific proteins, and they can be passed to the liver to have biological functions. So you see what I mean? 就是大家不好听，就是说你如果有兴趣的话，大家都可以看，但是要小心，因为 Vesicle 是一个坑。很多老师跟我说，我不想做，你们这个 characterization 真是有够复杂。我必须说这个是真的呀，因为我们连我们自己做了10年以上的 Vesicle， 因为刚才时间上的关系，我比较没有讲。可是真正的 characterization 都是在这些的后面，我看一下。Yeah, sorry, this is on my. Yeah, 这些都是 characterization， 因为时间关系我不讲，但是这些都是 reviewer 会跟你要的。Yeah, 所以我会跟大家建议，如果有兴趣的话，有一篇 paper 叫 MISEV 2018 MISEV。Yeah, 那篇 paper 是现在很多在做 Vesicle 人都会参考的一个 standard。Yeah, 然后你从那边的话就会比较少遇到 reviewer 的 questioning。
我请问一下，像那一些 G-loop、N-loop 这一些，它在小鼠或人本来身上是没有的嘛？所以如果用进这种生物体之内，它会产生免疫反应吗？
Very good question. We don't know about humans， 因为我们不敢拿人来打。现在想过学生可以没有开玩笑。但是我们在小鼠实验，就是在 immunosufficient 的时候，我们有做那个 immune panel。没有就是 Vesicle 载有我们的 reporter 注入进去之后，没有引起发炎反应。我们只敢这样子讲。So it's not too surprising in a way， 因为像 GFP 被用了这么久，就是它在小鼠也没有造成很显著的发炎反应。那我们是把我们 decorated 好的 Vesicle 注射到小鼠，然后去看它的就是 inflammatory index。没有那个很大的一种上下，就是差不多跟 marker 差不多。但是那个 data 我们没有用到这 paper 里面去。Yeah, but maybe that's something to consider. Yeah, 其实高效 bioluminescence 其实一直以来都稍微被荧光蛋白这一组群盖过去，但是 bioluminescence 在活体的影像上有非常大的优势，因为它自己会发光。对，跟我的名字没有相关，但是它自己就有内源光出现，它的 signal to noise ratio 很好。Yeah.